the last day for the ATAL FDP Faculty Development Program. We have with us today Professor Laddha sir. So to begin with, I request our co-coordinator for ATAL FDP and IQAC coordinator Professor Hilaker ma'am to welcome sir.
So please come on the dance.
Yes, photo session.
Yes, yes.
Thank you.
I'll just keep it here.
Okay, awesome sanctum.
Yeah. Sir, please. Please be seated. Please be seated for 2 minutes.
Okay, as per sir's, I honor sir's request, and since he has told that he will be introducing all of you to him with his work, I'll just quickly give a brief of what sir is going to talk on today. Sir is going to discuss on the botanicals of phytoconstituents, what are the opportunities and challenges that are available in that field. So, with all due respect, over to you, sir.
Thank you. Thank you very much. Somebody will have to operate for me.
Yes. Sir, I have the
You do that?
Yes.
Yeah, please, yeah. Yes. Okay, fine.
Madhu, handbag. You want handbag?
Oh, no.
Sir, can you? On this.
Small group. Not a big issue. I'm used to that because in ICT when I joined it was only intake of 18.
Yes.
And then it became 30.
Yes.
I hope I'm audible?
Yes.
Okay, good. Good morning to everyone. And I'll take care. Okay. Laddha from UDCT, then it became UICT, and now it is ICT. Okay, so we have transformed actually from what it was under Bombay University or University of Mumbai, and then we became actually institute, and now we have a separate university. We are a separate university, and there are two words actually in our institute name. One is chemical and technology, and I'll be confining to those two words, chemical and technology, through natural product. Okay, and that is my introduction. Actually, I'm there for almost now 30 years, 30 plus years. I've been working in from Rognosy. Probably people do not like this word. Okay, or herbal drug technology, that is slightly better. So I'll be dealing with all those terminology. Anyway, I remained confined to my field, and the area which I was catering actually is more extraction, isolation, and thereafter I'm making use of those material actually for making. That's it. I'm sorry, but yes, if I had to ask what is the aim, actually, I think the only aim of everyone actually is to make money. Primary aim. Secondary aim can be anything, it is like this, I'll put it actually, I will have to define that actually, making money actually in a fair way. Because we are all educated people and we would like to make money actually by our intellectual capacity. Or precisely I'll be dealing with that, and right from the day one, I must tell you, since I came from the industry and joined academic, right from the day one, my focus was very clear. When I used to start my day in the morning, at the end of the day, I used to ask myself actually what is my gain. I used to always, and I do this today also, when I start my day and at the end of the day, what is my gain? Either I should get in terms of money or I should get in terms of knowledge. These are the two things which ultimately it will be able to, you'll be able to convert into a money. All right. I'll be dealing with herbals, and I had a long journey in the herbals, so I have come across certain problems from the perspective of everyone. So rather, I will first start with those because I don't know how many of you have heard the good stories or the successful stories of the businessmen. They all started with the problem. Okay, they all started with the problem actually. If I had to, I don't know how many of you know the housing.com. Housing.com. Okay, housing.com was started actually by a young IITian. Okay, because IIT, he graduated, Yadav. He graduated and from IIT Bombay, job is not a problem. Got immediate job. Then the problem arises, where to stay in Bombay because he was not from Bombay. So where to stay? So he started hunting for a house. And then he realized actually it is a very difficult job. Okay, because he wanted actually that everybody should be able to get what he wants by sitting at home. And that is how actually the housing.com came. Before that, there was only one property portal, that was Magic Bricks, which was owned by Jain Group, Times of India Group. And housing.com actually changed the scenario actually to the extent because he was making you virtually get into the flat. That is how it started. Okay, and housing.com became actually the most, now I think the second one, most preferred site actually for, so it has come actually from a work of, the problem which he has faced. Gutka industry? Gutka. Okay, Kothari started first actually the Gutka. And he was the one actually who started with the Pan Parag. I'm talking about the Pan Parag story. Pan Parag I think is not very popular now, but there was a time when Pan Parag was very popular, and that was the first Gutka which came into the market, herbal product. And he was actually a vendor. So he was supplying all those which are required for the pan, like pan, supari, menthol, and then of course gulkan, all those things. And one day he got an idea actually, why not, instead of supplying all these in a loose, why not club together actually and come out with the gutka, and that is how the gutka came. Okay, so this is all the successful stories. And that is what, so I'll be starting with some of the problems which we face. All of us are facing problems. Okay, whichever subject you are in, problem may differ, but yes, we all face problems every day. And so I'm confining to the problems which are faced by cognizant people around, both student, faculty, institute. Okay. All right. So let me just go through some of this. They are very popular terms. Pharmaceutical botany, of course, nobody hears nowadays because initially it was named as a pharmaceutical botany. Okay. And there was one book actually by Yonke and pharmaceutical botany at university. We still have that book actually. And very nicely been made actually that book, and the classification which was followed was botanical classification. Okay, like families, order, families, those kind of things. But yes, he did not give much importance to the chemistry part of it because it was pharmaceutical botany. So that was a book which is, I think, now it is rare. For movies of phytochemistry, phytochemistry, yeah, that is currently people are comfortable with it because all of us I think all of you have referred these kind of books. Herbal drug technology is later added word. Okay, later added word because people wanted the technological touch to that, so they have added herbal drug technology. Natural products. This is the what do you call the word coin actually to club certain subject. Like pharmacology and cognosy club together and call it natural product. Industrial pharmacognosy, yes, today nutraceutical has come into the market, and industrial pharmacognosy is making noise now. And a lot of phytochemicals are now isolated, and quite a few of them have put into the commerce. Herbal cosmetics, today I don't think you come across any cosmetics which doesn't have the herb. Okay, and I think probably you will not like that. If you have a plain cosmetics made up of the chemical, I don't think people will buy that. So you have to have some herbal touch, whether it is there or not, that is a different issue. Okay, but it must have the herbal touch and some herbal name actually on the back. Herbal medicine, morning whenever I start TV actually, Madhunashini, and it is Quassia. I don't know anybody has seen that ad. Madhunashini ad comes, and it's Quassia. So any ad which is local, operator they give, and lot of herbal medicines have been advertised actually. Ramdev, yes, Baba Ramdev is a turning point. I'll put it actually for my subject. Because when I studied actually it was Ashwagandha root only was used. But Baba Ramdev came actually and he put the leaves also. Then how the subject got changed now, Anyway, so this is how the change will take place. Okay? Then herbals in healthcare. Well, today I think I'm sure all of you are taking some or the other forms of the herbal for healthcare. Okay? We start our day with a tea which is herbal. Either tea or coffee, so that is herbal. So I don't think anybody can keep away from the herbals. Herbals is an integral part of your life. Alright. Well, I'm talking about cognosy. You can think from your subject, whatever subject you are into it. Okay? And I know very clear that every subject is facing a problem. Okay? Subject, when it comes to the pharmacognosy, well, we have something which is close to botany. And the reason for that actually, because I was taught actually by the botanist, not by the pharmacognosist. So obviously when you have been, when you are taught by the botanist actually, there will be a lot of impact of the botany. And that is how the subject became actually very close to the botany. Today it is, I'll put it actually, I am transforming botany to the cognosy. Because I am on the board of many science college actually, and I am everywhere actually I am putting, because I am trying to impress upon them that your botany, there are no takers. And after graduating in the botany, actually what student will do? Nothing. You will have a problem with the job. So why not you start medicinal plant in that actually? So most of the syllabus in Bombay, in Bombay colleges, I must tell you, I have transformed them and made it more oriented towards cognosy. Medicinal plant. Faculty more oriented towards undertaking botanical and analytical work. If you look at the typical work which has been going on in cognosy, it's more botanical work like microscopy. I've got fed up with that. I don't know, I have put quite a few video actually on YouTube of the microscopy with the intention that nobody should do the year after the same microscopy. So that put an end to that, So botanical work, most of the people are still, even today I gave the thesis actually with a lot of this kind of description. I think this should be left to the botanist. They can do this medicinal plant microscopy. Analytical work, yes, because, and that too it was confined in most of the cases to the HPTLC. And I had a word with Ankuram once actually in Chavigaonkar, and I told him, you stop this free activity. Free activity, because he gives free service actually for HPTLC chromatogram. I said he gives the sample, he runs them, he gets a good picture and good chromatogram, okay, of which nobody knows what it is. Okay, so this is what, unless you run along with the marker compound. Okay, only mere fingerprinting doesn't carry any sense. Alright, so that was the work actually. So again, analytical work which is more repetitive. Syllabi have more analytical component and less technological component. I don't think we are teaching now. Of course, the new syllabus has now technological component actually, like extraction, the isolation has come into the picture, which I started of course long back in ICT. But yes, otherwise all earlier syllabus, if you see actually this part was lacking in that. Lack of required laboratory facilities, I don't think, I've been visiting number of colleges, and I'm sorry to say the status is pretty bad in the cognosy laboratory, because the teacher's orientation is also not there to that extent, and poor facility, at the most you have one subsolate. That is the only, sophisticated they call it. Things available. Alright, job opportunities in the field, question mark. Lot has been talked about it, but reality is far, far different than what it is. Though every company is working in the herbal, but the opportunities in the herbal sectors are still a problem. Course content does not create confidence to start own venture or be a self-employed. That's another problem, that the course content is such that students don't get that kind of confidence that yes, I can start after graduating, I can start something. Okay. And there are a lot of other factors which also contribute to that. I remember actually when I started my career, actually, as I told you, I have always wanted how productive I can be. So as to use my extra time actually for generating something. And everybody used to talk actually, business you need money. I said, I know, I don't need money for the business. What you need is a good idea for the business actually. Because I came here in Bombay, my father bought me here way back in 1977, and he took me to the market and introduced me. Look, beta, I don't know how many of you have seen the main market of the Bombay. Then of course the fellow used to have around a 6x6 one room, one revolving chair, table and phone, because phone was a luxury then. Okay? And he used to tell me actually, look, this fellow, typical Gujarati, I'm sorry if anybody is Gujarati in the class, it's a compliment actually. He said, okay, look, this fellow is doing good business actually in this room. Trade typical. And I got impressed then actually that yes, beside this, I have a this, which most of the typical traditional Gujaratis do not. They only buy and sell it. It's a trading actually. Okay, buying it actually at the cheaper rate, adding some profit and selling it, reselling it, because they have capacity, financial capacity to buy that actually and keep it for some time, holding capacity, and then of course as the market improves, they will offload it. Better. So I said I have a brain in addition to that. Okay, so that was the case. That is how I started actually. So yes, that was another thing, that people think that you want money to start the business. It is not so. Disconnect between academic and industry. This is another thing which has there's a huge disconnect between industry and the academic. Most of the teachers, I'm sorry, I'm also a part of it, but they are in their own world. Their ultimate goal is paper and patent. That's the end of it. Okay? Am I right? Everybody would like to have some paper immediately because all your further promotions depends upon that. Okay, papers and then of course patent, whether it is good, bad, but yes, you must have some patent on your name. Okay, whether it is having any taker or not, but then I think this are some wrong parameters which we have imbibed in our society and we are working for that. Instead, actually, I started my career, I went to the industry, I said, you give me only problem, I don't want any money from you. That's how I started. I said, you give me 10 problem, in case if I solve it, after solving I'll come back to you and then you give me the money. That's how I started my career and I never had any problem actually of the money. Just now when I was sitting there, I got actually, email from a US person saying that I want to contribute 5 lakh rupees to your lab as a donation. Okay, because we were associated with some work. Yeah, so I must tell you, he's a Maharashtrian, Ram Agarkar. He worked in the BMS company. I don't know, anybody know BMS? Okay, he was working, he's a retired person now. And now he's 86, so he become emotional now. Yeah. So, and we had a lot of association for one industrial project. And he said, I would like to contribute. So, I told him actually whichever we want to contribute, he said I would like to specifically contribute to your lab. Because he also started his career actually in industry in Herbal. And he was associated with the Taxol actually a story. Okay, Ram Agarkar, you'll get a lot of reference of Ram Agarkar actually in terms of the Taxol. Alright, so that was the case. PG level, postgraduate, that means master's level and the doctorate level, what is the scene of the cognosy? Research projects are more of generating data and lack of newer approaches. If you see the typical work or the thesis, is generating data. I have worked on Withania sominifera, then these are the parameters, somebody to replace Withania sominifera, start working on the asparagus racinosus, same parameter. So what you are doing is actually you are generating data, there is no newness in that. Okay, same parameters. I have put here actually antioxidant study. You take any herbal drug, any herbal paper today. For the last, I think almost 15 years, I am fed up with this antioxidant activity. Every plant is having the antioxidant activity. I don't think they need your validation for that. They have that. They are sustaining in the field. That is the reason. But yes, that is what we are doing it actually. Again and again generating the same data. I think we should stop. Instead, one should be courageous enough to start something new, even if it fails. At least you enjoy that. You have attempted something new. Okay? Research undertaken is more assorted and does not lead to definite or concrete outcomes. Everybody works in every field. Okay? Biotechnology, nanotechnology. The new word comes, actually we immediately fall prey to that and we start writing project on that. Buzzword. Because you also know that if I write UGC would like to have these words now. Then only the project will get sanctioned. But I never did so, I must tell you. I never did so. When first time for the project, I must tell you my story and how I was adamant actually. So I went for the UGC and I presented, okay, I will be doing the extraction isolation. And one gentleman, I will not name, asked me actually, after that what will you do? I said nothing. I said bluntly nothing. After extraction isolation, I will do nothing. He was expecting that, okay, I will put to some tissue actually, tissue will flicker. And then I will draw some confusion, something of that kind. I said nothing. And then I argued with him, actually, probably you are not doing any work of this kind, and you don't have any idea what kind of effort is required for this. And I said, if that would have been the situation, if everything would have been so easy, by this time the FDA should have had all the phytochemicals with them, actually the marker compound, which people would have used for that. Okay, so I must tell you, my project got sanctioned on the spot. Because other people realized actually, yes, this is the reality, and it was sanctioned on the spot. So sometimes don't be afraid of this evaluating, people actually, because they are also from your group only. Alright? Okay, so research undertaking is more assorted. I think one should confine, whatever area which you like it, you should confine to that. I must tell you, right from the beginning, I confined myself to the extraction isolation. It's not that I have not done other things. I never paid attention to that because I have to do. Microscopy, yes, I had to do microscopy for the undergraduate because it was put in the syllabus. Okay, analysis I have to do. But I focus only on extraction isolation. After extraction isolation, I close my mind. Okay, so I remain confined to that. And that is what I would expect that people, teacher, they should get into a very narrow field. And I must tell you because I referred one book of the flavonoid by Wilson. And lifetime he worked only on the flavonoids. And that is how he became actually some established person whom you can refer actually an authority in the flavonoids. And that is how it should be. Problems undertaken are more theoretical and are far away from the reality because we are not solving the industry. In spite of having such a huge wealth, I'll put it. Way back in 1998, when I started as a career in the research, I called a meeting of cognoscists in the Bombay because that time there were 4 colleges, main, ICT, BCP, KMK, and SNT. So I had a meeting in my institute actually about the 4 faculty, 6 faculty because we were 2, and the KMK had 2 faculty, and one faculty from BCP and the KMK. So I said, we will join together. So at any given time, we will have at least 6 supervisors in terms of research guide, at least 20 students because those days cognosy was quite prevalent. Actually, every college had a cognosy student. So at least 20 students will be working under us for the graduation program and 4 labs on at ICT, KMK, DCP and SMBT. So why don't we come together and then we go to the industry actually to start, say that okay, you just give us the problem and we meet once or twice in a year, take stock of the situation, give the problem. Because what I was seeing actually, my student is also doing extraction of the curcuminide, KMK student is also doing the same job, BCP student is also doing the same job, and there is a time limit. Master's program has a time limit. By the time you come to that, actually, there is no further work can be done. So it was repetitive in only in Bombay itself. So I realized actually we are putting money, but the science is not getting further developed. So that was my intention, but unfortunately it did not work out because I was the junior most done. Okay? But well, I made attempt actually. So to that extent, I like it actually. At least I am made. So then, of course, I started my career, alone. Let's, chalo, okay, akela chalo, like that. But that was the case, and I will still request in case it can form a sort of group. See, when you go to the industry, it becomes formidable group, unless you will take you seriously. Okay? So that is what is required, and this is what we are not realizing actually. The industry is facing a lot of problems. Many problems they are facing it actually, and they want solution from you. Okay, just now actually I have transferred a technology to a Godrej for extraction of the lawson, and there was a filtration problem. They came to me, at least they came to me for 6 to 7 times that yes sir, we are finding it very difficult for the filtration. So filtration was the aspect which is coming into the way of developing technology. So you can imagine the importance of the filtration, because henna leaf is very, very mucilaginous, and that creates a problem for the filtration. And our extraction is in an aqueous medium. All right. So both faculty and scholars are afraid of trying new approach, fearing that it may not yield desired outcome. Everybody wants stereotype pattern that you should be able to publish. And if you say, okay, sir, I'm not getting, I don't think guide will like this. Correct? No guide will like. If you are starting with the extraction of say vitafilin, and after putting in 3 months, you say, sir, I'm not getting the vitafilin A. Would guide like it? Tell me. Guide will not like it. I like it. I would ask what effort you have made. What effort you have made. Then I will ask, okay, if you have made this effort and if not getting, okay, we'll go back actually, we'll see for the property of the vitafilin, how we can get it actually, and I will monitor where it is. If it is not coming in my extract, where it is going. When we started with it. And you should not be afraid of trying something new. Okay? Both faculty should have either basic research or the applied component. We are in academic institute, and I know fully well. Okay, so I am not a researcher. Because Government of India has established research institute like NCL, RRL. They are 24 hours research. We are actually a teacher, so we have a limited time. And in that case, actually, I don't think we can get into any fundamental of the basic research. What we can do, we should be helpful to the industry or to the society by way of actually applied component. That is what we should be more concentrating, whether we can solve some industrial problems so that our work is more relevant to them. So applied, I always concentrate on the applied part. If you see whatever the paper I have published, those are not pertaining to any new compound which has been isolated. No, I have not isolated any new compound. I have only isolated in a feasible manner the compound which is already known and made it available at a very affordable cost. Because what was happening, it was not becoming actually the feasible, and with the result it was the major deterrent factor which was coming in the usage of that. Next slide please. Alright, pharmacy, what industry wants it actually from you? Technical skill, yes, you should be having some technical skill. I don't know how many of you have referred the book called Thin Layer Chromatography by Wagner. How many of you have referred Thin Layer Chromatography by Wagner? I think you must refer that book actually, of the natural product. And that book is only Thin Layer Chromatography by Wagner. And today that is the book actually which has been used by all herbal people, all. So the kind of the work which they have done and which he has done is absolutely of the meticulous and very pertinent to the usage of that. Good communication skill. Yes, you must have a good communication skill, because unless you have a good communication skill, you will not be able to communicate what you exactly want to communicate. Okay? Just to be on the lighter side, I am not a Hindi speaking person, I am actually a Rajasthani. And when I joined Mumbai Vidyapeeth, so if you, that time actually, because Mumbai Vidyapeeth, the entire staff is my and large is a Maharashtrian staff. So whereas to go there actually, and if you talk in English or in Hindi, the answer will be, if you talk in English, the answer is okay sir, we'll look into that and he will keep the file aside. Keep the file aside. The moment you say, sir, maza kaam hai, then the thing gets clear very fast. So this how it has helped, no? It helps. I have to go to the BMC. And the moment I have to take sahib maza kaam kara hai, and immediately the work is to get done. The moment you start conversing in the English, then the outcome is actually, okay, we'll look into that and they will keep the file aside. Okay, so the communication skill matters a lot. Time management, many people complain that they have no time. Teacher cannot complain that. Okay, teacher cannot complain that because teachers, the maximum workload is only 16 hours per week. Okay, maximum workload. Am I right? Has it been increased now? I don't think so. Okay, huh? It's a 16 hours or 18 hours? Okay, I'm sorry. I'm sorry. Even today I'm taking more than 20 hours. But yes, 18 hours is the limit. Okay, so if you take 18 hours actually and divide by 7, or even if you take it 6 or 5, I think it doesn't come out more than 4 hours per day. So teachers have enough time at their disposal. So I don't think time management, only thing that you have to effectively use your time, most important. Effectively use your time. I remember in cognosy, we have an experiment called ash value. And I'm sure some of you must have performed that. How long it took? Muffle furnace. Correct, no? You keep it in muffle furnace. Alright, so muffle furnace and the practical is this week, or whether no, if the practical is today, student will never come back to see the same practical. You are keeping the muffle furnace and forgetting that. Am I right? Because muffle furnace takes 4 hours. Okay, I never did so actually. I found out the ash value in 20 minutes. By Bunsen burner. Okay? Within 20 minutes you get the ash value. So that is how you have to, and I will rather appeal to all of you, though, I mean, since all of you are teacher, I think you should roll your sleeves and start working in the laboratory. Because only by working in the laboratories you will be able to bring the change. Otherwise you will not be able to bring the change, I must tell you. And that is what it is very important that along with the student, come out from that phobia that you will not be able to demonstrate. Come out from the phobia, sooner the better. Sooner the better. It's not that even today when I demonstrate, I do make mistakes sometimes, but I learn from that. Okay? So it is very important that unless you work in the laboratory, till such time you will not be able to find out the problem, nor you will be able to come up with any develop this thing. When I said actually now I am doing with the 20 minutes, this is because I realized the problem associated with that and then found out the solution for that. Okay? Alright, time management, critical thinking and problem solving. That is what when you work in the laboratory, you start thinking about it, why you are not getting it. And once you know why you are not getting it, then you start thinking. And I agree, whenever I don't know, those who are ICAT and they should be knowing it actually, I visit to my research lab at least 20 times in a day. At least 20 times. And I see students what they are working at actually. And when you repeatedly see, and I also work in the laboratory, and when I see actually something is going on, then I start thinking, at home I may be watching some TV, but in the back of my mind, the thing goes on. Okay? Teamwork and emotional intelligence. Intelligence actually teamwork. Yes, I remember my first student who undertook doctorate program under me. I said you'll be doing nothing but mounting column. That's it. Nothing more than that. So he was working 6 months for the column, and he isolated 6 zones, which was very good work actually, and it was published also very nicely. And we converted that project into the commercial project as well. And he used to come, and I must tell you the good story about that student, Dipankar Ghosh. And he used to come, he was married then, of course, and he came to me, and I gave him a free hand that I said, I will not be asking you when you are coming, when you are going it. So he used to come in the morning around 9 o'clock, work till 1. Thereafter, he used to go for lunch, have his nap, and 4 o'clock he used to come. Okay, I knew that everything. I never asked him actually. But after coming 4 o'clock, he used to work till 10 o'clock. Okay, so that is the case, and I think the guide must understand this. I gave him a free hand. Yes, you go for it. You want solvent, go for it. Whatever solvent you want, don't bother about the cost part of it. And that is how he was able to, because he got the idea that okay, nothing will come in the way if I work it, and he was working very sincerely. And by that one student, the good culture has been laid down in the laboratory. Yeah, that yes, yeah, he was working very coolly without conversation with anybody else. He was working. Emotional intelligence, yes, I think we all are having that. Digital literacy, I think you must have a good, and I think you have, I don't doubt anybody. Okay? You are more, internet and what do you call digital savvy today. I'm also, I must tell you. For my age, because I don't carry cash much actually, because I deal with I think you are having the GPay and all that. I don't have that, but I carry a card. Okay, and I don't think I have signed check in the last couple of years. I have not signed check, and I told my children also, even if I died, actually you can continue to withdraw money from my account. Okay, ability to take initiative. I think you must take initiative. Most important. Some problem if you realize actually, because I joined Cognosy, and I must tell you, when I joined, actually my contemporary said me, Laddha has gone mad. He has opted for the Cognosy because during my time actually in PhD, it was walking for any subject. When I got into the PhD actually, in fact 4 teachers called me actually, whether you want to join physics, chemistry, Cognosy and ecology. This was the choice to me. I said no, I will confine, and I was the first one of my guide. Okay, so the Cognosy, I said no, I would like to offer the Cognosy. And well, today whatever scene is there, the Cognosy, same scene was there then as well. Okay, intensity may be little less because today we have a volume. That time there was a less volume. Okay, in terms of manpower, in Cognosy. But the condition was same. But I said no, I would like to continue in that subject because I was interested in that subject. So and if also would have continued in the same mentality, well, that the Cognosy is not something which is a good subject, I don't think I would have been able to make that kind of impact. Okay, so sometimes it is always better that there is no much of rush. You should get into that line because the competition is less. Okay, bridging the gap. So these are all the government comes into the picture because government money is required because we are exploring, and normally the industry do not give the money for the exploring work. So government money is required actually. I will call it actually seed money. So government can start the work. Institute faculty, we must make use of that actually for the productive work. That is what industry comes into the picture. We must be useful to the industry. Whichever subject you are into it, forget about the herbal part of it. Government gives the money actually for the project which industry cannot fund. So you must actually work in such a fashion that whatever project you are working on that, at some day it should be useful, the finding should be useful to the industry. That should be the constant thinking in your mind. Student will also enjoy this. If you come to my lab right now, you'll find every student is working on some industry project actually, and all of them are going to the industry and their products are being sold. That they know very well. So they are all interested actually in working whatever topic is being given to them. Okay, so they will also enjoy provided you give them that kind of opportunity. Next slide, please. Well, this is about the faculty to which I also belong. Okay, faculty incorporate both science and technology component in their coursework. Basic science. Yesterday I had a meeting actually of the RGSTC. RGSTC, some of you must have put your proposal also. And then down they have one separate committee actually for the medicinal plant. And one thing I impressed upon them, don't get into the cultivation and don't get into the formulation. Cultivation is not our cup of tea because it's agriculture. And formulation, I said there are enough formulation in the market. And whatever formulation you put it, I don't think it can have an impact. Because there are certain limitations. Okay? So instead, concentrate on science and the technology. But well, I will leave it to that. They should supplemented with appropriate practicals with the scope for developing technology. I don't know how many of you demonstrate in the laboratory to the student, even if those who are working in the chemistry, whether you are able to show some 50-gram synthesized product. The answer is no, by and large. Am I right? Maximum RB in the lab will be of 100 ml to 100 ml maximum. Correct, no? And you need magnifying lens to see the product. In majority of the case, correct? You need magnifying lens. Alright. So that doesn't give that impact to the student that they are developing technology. In case if you show some product actually in a beaker or maybe petri plate actually a good quantity as a heap, then probably it will have an impact to the student actually, yes, it is possible. So you must have that kind of, so the practical component should have that, even if it is not for the individual practical, but for the demonstration purpose. You must have that component. I tell you, when incorporated 20 years back, I incorporated one practical actually, that separation of the starch from potato. I put that actually, and I took 200 grams of the potato, and from that I isolated around 30 to 40 grams of the starch. It contains around 17% starch. So 250 grams I took it. And when I started with the potato, and within half an hour I was able to demonstrate them starch. Okay, that almost 40 to 50 grams of the starch. And one of the girl student, she immediately said, "Oh, good sir, today even if we don't get the job, we'll be able to make starch and sell it." See the feeling. Look at the feeling actually. And that kind of feeling they must have it in the lab. Not just coming, routinely and doing something actually, and showing to the teacher and that's the end of it. No, that is not the purpose. The purpose is to imbibe that technological component into their mind. And for that, you must take effort actually. Okay? Even if you have to pay something actually, I think you must find out some amount actually and give some demonstration to the student by which they get that feel. Okay, this is a technology. Should be able to address industrial requirement in terms of analysis, technological aspect, or development of production. Yesterday, Himalaya called me. People from the Himalaya, they called me actually. Besides teacher, I am also a businessman. Well, officially not, because officially the business is owned by my wife. And she's a commerce graduate. She doesn't know anything about, even if I ask gallic acid structure, I don't think she knows about that. Okay. And my son is now operating, and he is actually MBA, he's pursuing his MBA. But my mind the brain behind that is me actually. Okay. So Himalaya called me yesterday. Actually, they would like to buy phytochemicals from me for the analytical work. Okay, so the company like Himalaya, last couple of months actually, I have sold consignment to the Saudi Arabia, to the Malaysia. So that is about the phytochemicals. Okay, and I must tell you the business. Anything which you export it is 100% tax free. Okay, 100% tax free. So you get a good amount of. Otherwise, I am in a 30% slab. Okay. Syllabus should be technology oriented, wherein phytochemistry and the chemistry, when it comes to the commerce, this is what is my view. You may differ from that. But I feel actually the commerce should be, more concentrated towards phytochemistry and the other chemistry part of it actually. I still enjoy that. Next slide, please. Research guide. Now from the guide's perspective, what guide should be? Out of box thinking. Don't have a stereotype pattern. Okay, you get impressed actually by what others are doing it. So before you start your training, what will you do? You'll go to the 10 people, what they have done, what they have been doing it actually. And then you get impressed. In my lab actually students are not, they are not supposed to be referring once they start the work. That is a culture. You do the reference before you start the work. But after you start the work, don't refer. Because once you start referring, then you do only experiment. Okay? And I don't see anything. Only book which I see actually while doing extraction isolation is the Merck Index. That too only for the property. Otherwise, I don't get impressed by any paper. Okay? I don't see any paper. I don't. Some people, those who might have seen me actually working in the laboratory, I don't think any time I refer any paper. Okay? I first read about the properties and all that, and then I make up my mind actually what is the other constituent which are present in that, and I make up my mind and start working on that. Okay? So don't get every time start referring referring papers and all that. Out of box thinking. I don't know. I mean, I don't know whether you're keeping track on the right side. I must tell you this product, Morning Fresh. And this was the product actually which was started by a daughter of one of my employee, and one of my employer rather earlier. And this was the product actually which is to overcome the hangover. Overcome the hangover. This is typically from the Bangalore. I don't know how many of you know the Bangalore culture. UB started from the Bangalore. UB, United Breweries. Okay. UB started from the Bangalore actually, and in Bangalore the culture is actually evening is a boozing time. Huh? And that is how the Morning Fresh came into the mind. No? Morning when you get up, you should be fresh. Alright. Sort of box thinking, project should be industry oriented or deal with the problems being encountered in the field. Find out the problem actually, go to the industry, discuss with them what are the problems which they are facing it, and if you can make a little difference by solving those problems, I think you will also enjoy and the industry will get benefit. Instead of working in the diverse field, concentrate and pursue specific field, whichever you like it. Okay? I will leave it to you. I mean, that is how I started. That okay, I will be confining only to the extraction isolation, but yes, only then you can, if you pursue for a long time, only then you will be able to bring some change in that. But if you keep changing your speciality every often, I always tell people actually in the industry the culture is every 3 years people change the job. The reason is because after 3 years people start expecting from you. First year nobody expects in the industry because you are new. So they know actually first year you will not be able to contribute. So they understand okay you are only getting to know the things and learning it. Second year they starts. And third year onwards they expect that you must have some output. And when the people cannot do that, then they change their job. Okay, so this is the pattern actually which normally goes in the industry. Alright. Scholars must become expert in the specific skill. When you undertake student actually for your project, at the end of the project you must ensure the outgoing student must have developed some skill in some direction. Most important. Okay? Today's student doesn't know how much quantity is to be weighed actually for undertaking the analytical work. That is the reality. Okay? And many students do not know how to even handle the balance. They'll put 50 milligram, then the requirement is only 20 milligram, they will take it out. Okay? I mean, you can see this actually is happening, everywhere. So there's nothing new in that. There's nothing like positive or the negative research, it is outcome. Understand that. It is outcome. All my students actually, the degree is guaranteed on day 1. They are done with the degree because examiner is my friend. As simple. Okay? So none of my students actually are worried about degree part of it. You can ask any of my students, they are not worried about the degree because they know they will get the degree without any problem because that is my job. Okay? What I expect from them actually is something which is new. The challenge which has been given to them that they must solve it. Alright, next. Syllabus should address, this is about the syllabus, identification of the raw material in our case. I must tell you one story actually. I went to one trade show in the US and the APA, there is a American Herbal Product Association. APA is a organization. And they have started making actually pharmacopoeia, American Herbal Pharmacopoeia. And they have started developing the monograph. And it is there on the website also, APA and the monograph. And if you refer one monograph, they have given a couple of monographs which you can refer it, others you have to buy it. And each monograph is more than 30 to 35 pages, each monograph, understand that. Very meticulous work. Okay? And I was sitting in that meeting actually, and Roy Upton was a person actually who is doing all these things. I said, your monographs are useless. I said, your monographs are useless. He immediately got irritated with me. In what way? I said, industry doesn't have time to turn the 40 pages. Industry want actually 2-page monograph by which they can confirm and ascertain whatever raw materials they have, it is right or wrong. Number 1. That means identity. And second is the purity. That means the constituent which are likely to be present in that, whether they are there in that concentration or not. This is the only requirement of the industry. They don't have the time actually to turn 40 pages and see. This is academic, we can do 40 pages. No? Academician, we like a thesis actually 500 page. Correct, no? More the voluminous thesis actually, which okay, so might have done more job. So that is the case, Extraction and isolation of phytochemical case studies. Around 2 years back, actually during COVID, we have published one book actually on extraction and isolation of the phytochemicals, wherein we have presented 25 case studies. And this was the challenge which I have thrown to my student, and this is the outcome actually for the last some 10 years of job. So I have given each of my students actually the assignment that you refer the extraction and isolation process, what is been published, and same process I want you to make it actually one which has been mentioned for the 2 days, I want it in 2 hours. That was the first challenge. And having achieved that, because quantity was not a challenge, quantity was not the intention. I wanted whether I can demonstrate this experiment within 2 hours. Because I know the psychology of the student in 4-hour practical. I don't know how many of you have studied this, but I have studied very deeply this psychology of the student. That first half an hour they are very active because they know what to do. But when they start the work, and in first hour if they don't get anything, then they are not there mentally. Okay, they are elsewhere. Physically they may be there because at the end of the day you'll give you'll sign it, then they will mark it, those kind of job. So that is the case. So I have thrown the challenge to my student that whatever method is there, see whether you can do this within 2 hours. Having done that, then we have said, okay, now whether we can further optimize and make it to 1 hour. So like that we kept on doing this from what it is mentioned 2 days, 3 days. We have been able to do the same job actually. Now today we have a method published in that book actually which you can carry out and demonstrate within 2 hours and 3 hours. And I don't know how many of you know, I run extraction and isolation actually a workshop. Our last workshop was in the month of August actually. And there we demonstrate almost 13 to 14 case studies that from the extraction isolation to the semi-synthetic and even synthetic. In 2 days, almost 15 case studies we undertake that. This is to build the confidence of the people actually to get into the extraction and isolation. Or at chemistry, biosynthesis, semi-synthesis and the synthetic analogs, I'll be touching upon that to some extent. Those are products, some of the, this is of course senoside, then you have adenine, and then dicin. I don't know how many of you know dicin. Dicin is an anti-arthritic drug. I will come to that little later. Formulation development, yes, that is the area. I'm looking for a formulation development from the academic institute which work for sure. Formulation which work for sure. I'm talking about the herbal formulation. Paracetamol tablet, you take it and the temperature gets reduced within half an hour. Okay? I mean, that means the effect you can see. You tell me whether you can get the same effect from any herbal formulation. And that is why they say, okay, you'll take for maybe a month or 2 or so. Okay? So I think there is a need actually that we must come out. See, now I'm not expecting in every field, but at least some field. Let's say talk about the cup syrup. Cup syrup, we must have the herbal cup syrup in such a fashion that yes, the moment you take it, within 15 minutes, you will start getting the relief from that. So develop such products which is required. See, the current generation is very different, I must tell you. Very different actually. They do not like actually something which is lingering for a long time. So formulation development, there is a need to come up with a newer formulation. Recently actually in one of my products has come out in the form of chocolate, herbal chocolate actually. Okay, you can see the company called Erlang, www.erlang.com, and yeah, you can open and see right now, no problem. And we came out actually with a chocolate, herbal chocolate in a bar form. I'll be showing the pictures of that later. So formulation development, transformation, traditional formulation, the newer. Some of the traditional formulation are working, no doubt. As I told you, cough syrup is working. But how you can make it more effective? That is the challenge actually. Okay? And that challenge we must face it. But I have been requesting actually to the industry that you are putting Vasaka. How much you are putting, I have a big question mark. Okay, because nobody is monitoring what is the vaccine content in that. And as on today, I don't know if any pharmacologist is in the lab and if he or she can tell me what is the quantity of the vaccine which is required to give the soothing effect. We have not worked in that direction at all. There is a need to work how much vaccine is required per 5 ml. That is the dose which is recommended. Then accordingly one can have the standardized formulation. Analysis of the herbal formulation, big challenge. Raw material itself is a problem, so the formulation is very complex, very, very complex. Okay? But if you can do that, nothing like it. I will be presenting one case study on that. Alteration, the determination is rampant actually. I'm from the field, so at least 400 drugs I can identify and I can find out the problem associated with that. But I'm sure those are into the cognizy and even into Ayurvedic colleges. One of my vendors says, sir, you are having the problem, I'm having the problem with you, but I don't have any problem with any other people. Especially the Podar, we have a Podar hospital and they have a Ayurvedic. So the Podar student goes and asks for anything, that fellow says, whatever they ask, I have. So I asked him, how come? He said, it's very simple, sir, because they don't know. So whatever they ask, I give it to them, and one thing which I know very sure, even if they take this, they are not going to have any serious side effect. So thing continues. Okay, so this is the knowledge. Alright, next. Suggestion. Identify the problem which you feel comfortable to solve. Identify the problem, it needs to be discussed with the relevant industry. Ask the problem if they are encountering and if one can work upon it. Start with that. Okay? I don't think you will get outcome in first student or the second student. Have the patience. Okay? And call examiner of those who are your friend. And make matter simple. Okay? Because we are all sailing in the same boat. Okay? So understand the problem of each other actually, and because ultimately we have to change the field. Nobody else is going to change the field. Understand that. Okay? I don't know. How many of you signed on the journal? Requirement. Correct? I have not signed for the last 30 years. And my suggestion is actually why sign when you are copying it? If you cannot copy properly, am I responsible for that? Huh? Think about it. And impress upon. See, sometimes I think you should bring this change in the society. It's very important. If you're all teacher actually, whoever is coming is another teacher. I don't think you are getting an inspector actually from some other field. He is also teacher. Okay, save your time and utilize your time actually for the better work. There's no point actually. In fact, once I had a problem actually with an examiner. He said, no, your journal should be signed. I said, what is the mark allotted to that? 1 mark, cut it. That's it. What is this nonsense? Think about it. What you are doing. I don't know why you are doing all these things. Are students interested in the marks today? Answer is no. Because nothing depends on this. We found nothing depends because thereafter everywhere there is a city. So think about it, bring about some changes actually, save your time and interest for the better work. Alright. From a small group of 4 to 5 people actually, I'll put it to you that those who are from the same, I think in every college, unlike in our institute actually, and specifically in the cognizy, I am the only one for the last 20 years. Huh? But otherwise in all institute actually, we have at least in every subject there are 3 to 4 people. Join together. See, more people, more ideas will be there. Okay, more money will be available. Even if you have to put it from your pocket, more easy money will be available. That is how most of the successful stories started in the ICT, then university. Most of the people, if you see today in the science and technology, they are from the university because while doing their masters in the doctorate program, they all joined together and started their work. One example, Galaxy Surfactant, the number 1 company today. The 4 people graduated, they started some activity in the Ghatkopar, one flat, and today they are Galaxy. So have that kind of thing actually, that form a small group actually, keep aside your personal thing, come together for a subject. Okay, everybody were ambitious, everybody has our own requirement actually, but keep it aside, come to the subject actually, join something. See, when you join 4 or 5 people together, the work can also be divided properly. Otherwise, you only land up by doing everything. Conceptualize, brainstorm, conceptualize, evolve feasibility or viability and start working upon it. These are some important things actually. And industry does this very regularly. I was working in the industry, I must tell you, FDC then, Fair Corporation, later on it became actually, I think the food, drug and the cosmetics of chemicals. Okay? Chandavarkar is a company. Electral is a product which all of you should be doing. Electral is the number 1 product. And I was working in that company way back in 1982. And I was working in the injectable section. Okay? Injectable section is the most sort of section. I was working in that. And I remember the company employed one person who used to come morning like this. And he used to see us. And whatever is going on, some autoclave is going on, some filtration is going on. And he would take a round of half an hour. Then go on. Nothing. An industry employed to him actually, whatever operations are going on here, whether you can optimize those operations. Because we were doing the routine work. So we are not going to change, we are not going to bring change. Because we have a problem actually, right from the top, the marketing people, they have a pressure, okay, this much output has to be there. Parimon, yes, you need actually 30,000 vials every day. So we had no other option. Start making Parimon. So our morning job actually go to the store, get the other, raw material, start working, autoclave, filling, once again, autoclave, do that, and that was the end of it. That's card. So they employed a person to see all the operations and whether you can bring the change. Today, chemistry people actually, even if you can bring change of 1% in the aspirin manufacturing, I can tell you, you'll be a blockbuster. If you can just optimize the production by 1% extra of whatever is happening today. Okay, because think about the magnum which is just produced, and 1% can make a hair of a difference. All right. Develop habit of working in the laboratory, that is what precisely I said. Is enjoyable? Is it? Okay, enjoyable, and will also be able to bring positive change in any experiment. Think of it. See what syllabus we're following, it actually has been set actually for the last 40 years. Same syllabus you're continuing, you're not bringing any change. So please start working, and you'll be the one who will be making the change in the syllabus. Okay? I must tell you, the syllabus which has changed in the cognitive to a great extent is because of me, because I started introducing actually the extraction isolation experiment. And today it has been gone into the mind of the cognitive that yes, this component must be there in that. So you must do some work and forget about whether they are takers or not. One day people will realize actually and they will start. Do not follow or copy others. Don't do that. Okay, none of my students goes and talk to anyone what they are doing it. Okay, start something actually on your own. Do the things which one enjoy, remain focused and pursue consistently. You are bound to become expert and successful both career-wise and monetarily. Do self-audit every day, most important. You must do audit every day, that whatever you start the day and what is the outcome in the evening, that you must monitor and do the audit actually. And if not day, at least a week, if not week, fortnight, every month, and we do that. Many times we sit in the laboratory and see what is happening, what is the outcome. We have not published paper for the last 2 months. So do audit actually. Okay, next slide please. Strengthening pharmacology, traditional system to medicine, unearth science and develop technology. There are a lot of scientific things which are there in the Ayurveda, a lot of scientific things. Extraction itself, when I see actually the extraction, Ayurveda use only 3 solvents: water, alcohol, and oil, taila. Okay, they never had these solvents. Dichloromethane, benzene, toluene, ethyl acetate, methanol. They had no. But with 3 solvents, they managed everything. Imagine, with 3 solvents. They have a product called, as it is, a kada, kashay, water extract. Arishta and asava, alcohol extract. And kaila is non-polar extract. Okay? And they use all the plant material very meticulously, even without knowing the chemistry of that. Understand, Ayurveda when evolved, they never knew the chemistry part of it. But in spite of that also, they use the appropriate solvent for those constituents. For tannin containing thing, they use only water extract, kadha, as simple. For asvarishta, ashwagandha is always either you take churna or you take ashwagandha rishta. Ashwagandha kadha is not, because steroids which are there in that, they are not soluble in water, but they are soluble in alcohol. That is why they are used arishta. Understand that. Okay, even without knowing the chemistry part of it, they still use solvent very meticulously. So that is about the unearth science and develop technology. One should not believe that only unprocessed natural products have innate superiority, and this is the typical Ayurvedic practitioners thinking, that they would like to have a proved product. Nothing. Ultimately, when you eat vegetable or the food actually, it's only something which can be digested, the body accepts. Something which cannot be digested, we know that we have no enzyme to digest the cellulose. So next day it goes into the fecal matter, as simple. So if you can remove those unrequired thing or the undesired thing, yes, that will have the better things. Be open and adopt systematically clone things from the other system. I think we should be open, not for, scientific thing from the other system also, but even plants I'll put it actually. The chocolate which have come out actually with it, there we have put even the exotic medicine which are existing in the US and the UK, because they were having a good scientific input in that, and that is how we use that. Develop scientific and strong process to manufacture the product to improve the health of the public. Manufacturing, yes, it is a major product. Many times you must have heard actually that the US the product goes, and then of course it gets rejected because it is having the heavy metals more than the prescribed limit, because they do not follow the good manufacturing practices. So there is a scope actually that yes, we must undertake manufacturing practices, impress upon the industry. I remember actually, I will not name the name of the company, so because I am dealing with a crude drug, so I know the problem associated with that, especially the one which is having the starchy. And in bomb, if I keep it after some time, it gets infected. The insect infestation starts in that. So I have just asked industry, how do you manage? You stock it up actually in a huge quantity, more than 1 or 2 tons of, let's say, glycyrrhiza. Glycyrrhiza is very prone actually for the insect infestation. The answer is, it's a very disturbing answer. कुछ नहीं, सब extract कर लेते हैं. You understood what I said? Even the infected one actually, they extract it and they think that nothing comes. Even the extract, poisonous thing of the insect will come along with the extract. Understand that? Okay? So this is the situation. So there is a need actually to develop scientific and the strong processes, publish scientific outcome without prejudice, don't manipulate. I have seen actually even the spectra is manipulated. And today we are in a better position with the computer around. We can manage the peak, get the peak. Am I right?
Yes.
I can be a free actually because I am also a teacher like you. Huh? So let us accept. I have seen actually in my own institute, I have seen, I don't know whether Jitu was there, Jitendra was there actually. His job was to, because now we get actually a spectra actually without in the printer. Those days were by hand. So the chemistry student used to go to him actually, and there should be one extra peak incorporated there. No, that was the case. All right. Okay, once you believe that, I am done. Okay, yeah, I think publish scientific work without any prejudice. Even if you don't get it, publish that, because what you don't get it is useful for someone. Somebody that, okay, this is the process I followed and this is what are the outcome. See, when I'm working on that, I will use this reference and I will not make the same mistake of going this way. I will rather think other way. Okay, so whatever work you are doing it actually, well, whatever work you do it, even if you don't get the output, but your approach should be logical, most important. It should be scientific and logical, and that you must publish. And I think I will even suggest the publishing authority, they must publish such work. Then only the effective change will take place. Quality literature, books, pamphlets, journals, and the internet. Today I don't think I have brought up in the culture of referring chemical abstract. Today I don't think any student refer chemical abstract. Go to the net. Okay. Even the seminar actually, they refer actually only the commercial site. That was the height of it. And they put only the one, half story, because commercial site will never say the other story. They will say only what it is be suitable to them to promote the product. And most of the time, students take out the data from there and publish it. I have seen actually how something which is having anti-cancer activity, it becomes drug within no time. In 2 seminars, it becomes drug. Okay? Having biological activity, in 2 seminars, it becomes drug. The third student says, yes, I have taken the reference, it is anti-cancer drug now. Okay? Alright, next. Alright. Technological development of the extraction and the isolation of the phytochemicals. I will deal with a few case studies. You tell me when you want me to stop. I'll do that. Huh? Yeah, this is well different product. Ashwagandha is available in so many different products. That is the complexity of the analysis. Okay? In herbal, you phase analysis because ashwagandha is available in the dry root form, ashwagandha churna, ashwagandha arishta, ashwagandha extracts and rest 1.5% bithanolides, betaferin. Now betaferin is available. I mean, many people are using it actually as a marker compound for the standardization of the ashwagandha. This is betaferin A. These are all the products. Next slide, please. Phytochemicals and different terminology which are used in terms of that phytoconstituent, secondary metabolite. By and large, phytochemicals are referring to the secondary metabolites and not the primary metabolite. Bioactive compounds, plant analyte, biomarkers and the markers. So these are the various terms which are used by the industry depending upon something which is suitable to them at that particular point. Next. Case studies, one plant actually, Electra parasitica plant. This of course work I have undertaken actually for a monograph for the ICMR. We have published around 60 monographs in the ICMR that was quality standard of Indian medicinal plant, and we got published I think in the 17 or 18 volumes, and we contributed around 60 monographs. And one such monograph was azafrine. I am sure I think none of you must have seen this particular plant. It is parasite. Okay, this particular family is actually meant for the parasite. And Electra parasitica, that is a plant which is a parasite on Vitex nigantu. Vitex nigantu is Nirgundi. I'm sorry, Nirgundi. Nirgundi is a plant, and now of course this plant is renamed. Now it has become actually Electra chitrakutinensis. Chitrakut. Chitrakut is actually a religious place in UP, and I went to collect this particular plant there, and it is found all. In fact, hill after hill you find actually Vitex nigantu is doing. And at the Vitex nigantu as a parasite, you'll find a tiny plant of 6 inch at the most. This is a 6-inch plant. Okay, this is entire, this is a rhizome part of it, which is appearing yellow. And this is the plant, and there locally people use it actually as a leprosy, for the leprosy purpose. Okay, so we isolated actually, these are all, well, this is about the phytochemical marker compound, sigma-star cytosterol. What is the difference? This is 18, 19, 20, 21 and 22. This is the only difference between here. One double bond. Okay? And I have seen some paper, people are trying to isolate sigma-sterol, cytosterol, it will not get isolated. It will not get isolated, I must tell you, because they differ only by one single double bond. So they have very similar properties. What one should do instead actually, look for a plant which is rich in stigmasterol like soya, rich in stigmasterol, isolate from that. And look for a plant which is rich in the background, cytosterol, just cytosterol. But don't try to get into that actual situation. Understand the problems. Quercetin, I am selling quite a good amount of quercetin, and I am generating only from the routine. Because, and I have seen number of papers actually people are trying to isolate quercetin. Please stop that. Okay, because you are not going to get actually very meager quantity will come. Instead, it can be easily converted because it's a glycoside. Routine is a glycoside. Okay, routinoside. It can be cleaved without any problem. In one single step chemical reaction, acidic hydrolysis you carry out, actually you get the quercetin generated in that. That's it. If you keep it for half an hour, actually the entire routine gets converted to the quercetin. Okay, so there is no point in isolating quercetin from a source which contains very less amount of it. Instead, convert it from the routine which is easily available in Liptograbilans, or even there's another plant called one more plant which has been used in the northeast. Okay, alright, so this is about requirement of the phytochemical is that for the analytical purpose, yes, it should be unique. When you use it as a marker compound, it should be unique to that particular plant. Like as I told you, azafrine is a carotenoid which is present in the Electra parasitica, which is a unique. Azafrine is not present in other plants. But if you take beta-saccharin or sigma-sterol, every plant contains that. So it cannot be a unique biomarker. Use marker actually which is unique to that particular plant, that is the requirement. Isolated from a plant. In case if you want this, there is no point actually in isolating, instead go for the semi-synthetic or even synthetic matter for that matter. Synthetic purpose. Next. But this is what my starting of business activity. Okay, I got impressed with this money. Gindamycin, 27,000 for 5 milligram. Okay, so that went 41,000 rupees. If I possess 1 gram, 2 gram, I don't think I need to work from the 8 to 6. Okay, Vasicin, 33,000 rupees for 20 milligram. I am selling at 3,000 rupees for 20 milligram. Okay, it may not be as pure as Sigma. Okay, Sigma may offer maybe a 98, I have around 95%, but I think that works. Okay, Bisaferin 35,000, argonic acid 36,000, and Plumbagin 9,000. These are the prices and very impressive prices. Okay, and that is how I started actually. That when I looked at it, actually, even if I get Gindamycin in small quantity, I think I'll be able to make good. I have one fridge actually, which is right now having more than 50 lakh rupees worth of material if I go by these prices. Okay, alright, next slide please. There must be some driving factor. Extraction, isolation of phytochemicals and the challenges associated with that. Yes, it is problem. It is not all that easy. Mindset, you need to have a mindset that yes, I would like to get into the extraction, isolation, whatever difficult job it is. Okay, you should be prepared mentally that yes, I would like to get into that. And it is a courageous activity, I must tell you. Facilities. Facilities cannot be a deterrent actually, and facilities can be acquired over a period of time. Okay, as I told you, Ayurveda managed with only 3 solvents. I don't think it needs facilities. Okay, so think from that angle, you don't need very sophisticated. Those who knows me actually in my lab, you ask any student of my lab, "Okay, how much they're interested in any instrumentation, and the answer is no, I don't even look at it. Okay, apna hath Jagannath. Okay, because I am confident whatever I isolate, I mean, instrument can only confirm that. Instrument cannot do any job for you. It can only confirm. Okay, HPLC will confirm what you have put into that. They are going to give the ideas. Okay, so use instrument actually only for endorsing what you have done. Extraction processes, you have to keep trying it actually, look at the molecule, what it is, keep trying it, think differently. I use all sort of things, chromatography, solvent extraction, chemical treatment, everything I undertake actually depending upon the molecule. Identification, yes, I am wrong. At the most, what I used actually to monitor my progress of the extraction is only TLC. And today I can run TLC actually, and you will get the TLC which I run it actually, it will give you an impression that is run on the instrument. Okay? All my students actually, they are very comfortable with the running TLC. Within 5 minutes, you will get the TLC result. Within 5 minutes. Okay? For all the solvent, we have a ready solvent in the lab. You come with any sample, within 10 minutes, we will give you a TLC. We have solvents for at least 30 different solvents kept ready. At any time, we can use it and immediately run the TLC. Purification, yes, it is a very exhaustive process, purification. I have undertaken forced calling, and normally what happens actually, the prices you think it is in a geometric proportion. Correct? If the 10% is 5,000 rupees, so 100% should be 50,000 rupees. Am I right? That's simple. Geometric proportion. In Herbal it doesn't go like this. If 10% is 5,000 rupees, 100% will be 3 lakh rupees. Because during purification, there are a lot of considerable losses takes place. And that is why the prices of the herbal, as you improve the purity, there are more losses, and with the result the cost goes up. Okay? Alright, so yes, time consuming process, it is a time consuming process. I must tell you, extraction isolation is not something actually that you start 10 o'clock and by 10:30 the product. Nothing of that kind will happen. Okay, sometimes I have to keep it as long as 20 days to see the product. So you must have that kind of patience. Frustration, part of life. Okay. So you have to overcome. Frustration is bound to be there everywhere, So you run it actually, sometimes the bigger breaks in which the product is being kept, the entire thing spills. Nothing you can do. Okay? So frustration is a part of it, but yes, you have to overcome, and every step you must learn. Every step you must learn. I'll just tell you one forceful story which has happened with me. It was in 2002. One gentleman, typically a Gujarati fellow, came, and it was during Diwali vacation, and he came to me, "Sir, I have one problem actually. I have given 2 days forceful route actually for the extraction by the CO2 extraction. Okay, supercritical fluid extraction. And initially he promised me that he will get the white product. Typical Gujarati fellow who doesn't know anything about the force-calling. He's a trader, he wants to sell. And that time force-calling was very much news actually. And he said, "Sir, he has given me now 120 liter of a extract, tari extract. So now I cannot sell this. And I cannot break my head for such nonsense, and he has invested a huge amount of money. The raw material, and that time the CO2 extraction was costing 60 rupees per kg processing charges. So you can imagine. Okay, what were the processing charges? Then he came to me, I said, "Okay, I have not worked on the force-calling so far, but if you give me some time and some money, I'll work it. Nothing else. I want only solvent money, nothing beyond that. And only if I develop, then only I'll come back." He said, "Yes, I'm ready to do that." So he paid me around 25,000 rupees unofficially. And he bought some 1 or 2 kg of the tari material to me. And then I of course looked at it, Merck index, and saw the properties. And I was impressed with one property, that it is soluble in methanol and chloroform, but it is insoluble in petroleum ether. And then I developed actually a technology by using this particular word, insoluble in pet ether, but soluble in chloroform with methanol. And he used actually during extraction, he used actually entrainer. I don't know how many of you know about the supercritical. Supercritical is not meant actually for the polar molecule. Phosphorus is a polar molecule, slightly polar molecule. So supercritical using carbon dioxide is only suitable for the non-polar. But if you want to make it suitable for the polar, you need to use entrainer. That means some alcohol in that. And that alcohol he has used actually 10%. So ultimately he got a tarry material. Otherwise in supercritical you don't get any extract without solvent. I mean you get the extract without solvent. Okay. But he got the with solvent because he has used the entrainer. And then of course I started working on that. First day I remember actually, because no students were not there because it was Diwali vacation. And first day I worked actually and then carried out some work. I was monitoring the TLC and well it did not work. But yes, it was showing the presence of the extract, that was the only case. Then second day what I did actually I removed the entire alcohol and I got tarry mass which I dissolved into the chloroform. Kept it like that so it was easily going into the chloroform. Then I added into the petroleum ether as it was told by the Merck index. Nothing worked actually. Second day I left it that way. Then I came actually and saw some sediment. I removed that sediment actually and put on the TLC and would get the concentrated spot. And thereafter I ran almost, I remember on that couple of days I might have undertaken some 50 to 60 a small experiment. And fourth day I cracked actually, and within next 20 days actually I remember I have given him 3 kg out of that tarry material, pure phosphorus isolated 98% purity. Okay, so I mean you take some time, but yes, frustration is bound to be there. One more my case study actually aloe. We were undertaking actually the trials actually in Tarapur. And we started around 10 o'clock, kept it actually for the oxidative hydrolysis using ferrichloride and the hydrochloric acid. And I was unable to see the product. We got frustrated. I said, let's go for a food and then we'll come back. So we went for a food. We're leaving it as it is for the heating. And when we came back, incidentally what was happening actually, it was in the lab actually and the light was flashing on it. Sunlight was flashing. And I could see all the red particles there. I said the product has come. So it was just actually because I was not giving the enough time to form. It was requiring 4 hours. And we were every time we were actually removing actually within half an hour, 1 hour. That is why the product was not getting formed. That it just so happened actually we got frustrated and I said okay we'll go for a food. We went to a nearby hotel, had a couple of hours actually for the food and then we came back. Incidentally we came back it was 4 o'clock and the sun rays were directly flashing on the flask. And I could see all the red particles which were, though the entire thing was looking black, but then red particle I could see. So I said, okay, filter it, and I could see the product aloe model, form into that. And then we developed the technology. Actually, we optimized and developed the technology for that. Okay, so that is all. Determination, yes, we have to leave it. These are all, I think, all of you have seen this. Atropine. These are all the plant products which are used as drug. Atropine, digoxin, leukocyte, sinar. Sinabine I will be particularly mentioning it. Sinabine is the only of the silamarine rather. Silamarine is a mixture. Okay, so pyranoflavonoid. Sorry, not pyranoflavonoid, the lignan flavonoid. Okay, lignoflavonoid it is. This is a lignan molecule. This is. So silabin is present in Silabin marianum, and as of today, this is the only drug which has been used actually officially proved as a hepatoprotective. There is no other drug which has been proved as a hepatoprotective. So silabin is the only drug actually which contains this is silabin A, silabin B together. They are called the silamarine. Morphine. Carbuxolone is from the glycyrrhiza, which was earlier used actually as a antacid. Reserpine, of course, is used even today. THC and CBD, I think most of THC and CBD are the buzzword now. Don't get into that. THC, CBD. All of you have heard this word recently. THC is tetrahydrocannabinol and CBD, of course, is cannabidiol. Diol, which is diol actually, and this is this comes actually from the cannabis sativa. Okay, and currently it has been talked about a lot actually, and now India is also permitting. US of course they are permitted now. Okay, US they have marijuana. Marijuana is another word for that. And CBD is found to be very effective now. CBD. So CBD very soon actually it will be cleared. CBD. Taxol. Next slide please. Yeah, these are actually phytochemicals as a functional food. Curcumin, we are the one, we are largest exporter from the country. Anybody knows actually lacadon turmeric? Anybody knows? Lacadon turmeric. Okay, lacadon turmeric actually coming from the northeast and that contains almost 9 to 10% of the curcumin. The curcumin, the turmeric which comes actually from the Sangli contains only 3 to 5%. The one which comes actually from lacadon contains 9 to 10% curcuminoid. Ellagic acid from pomegranate or from terminal chebula. Phosphorus, coleus phosphora, diterpene it is, Latin derivative. This is piperine from black pepper, I think a lot of product it is being put now actually, a lot of even allopathic product, there is some combination of the curcuminide and piperine, because piperine is found to be having the property of the bioavailability enhancer. Okay, so look at actually Ayurveda, how they are used, you look at any Ayurvedic formulation and you will find by default either black pepper or long pepper is there actually. And today we came to know that yes, it is bioenhancer. Lycopene from tomato, this is ellagic acid, hydroxycitric acid from the Garcinia cambogia, that is Ratambe. Anybody is from the Konkan, they should be knowing it actually. Dhirambe and Ratambe. Okay, Ratambe I think indica and Dhirambe is actually Garcinia cambogia. Mangosteen is a fruit which is from the Malaysia. So isoflavone, which are very close to the, if you look at the spatial structure of the genistein, which is very close to estradiol, and accordingly genistein is having the estrogenic activity. Estrogenic. Next slide please. Phytochemicals in the food sector. Annatto, Amul butter. Amul butter, the color is annatto, okay, which is used in that Amul butter. Lycopene is now currently been used actually as an antioxidant. Vanillin, of course, I don't think there is any ice cream without vanillin. Base flavor is vanillin in that. World's most popular flavor is vanillin. Vanillin, only 1% of the vanillin comes from the natural source. Only 1%. Okay, 99% comes actually from the synthetic source. Guaiacol is the root actually. Guaiacol is the root from which it has been obtained. Gallic acid and the gallate are popularly been used in the food sector. Propyl gallate, methyl gallate, those are the antioxidants which are used. Stearyl glycoside, I think quite a few of you must have been using at home actually by the diabetic patient. Stevia rabuliana, that is the plant. Tomatin is actually another sweetener, and it is from Tomatococcus danil, African plant. Okay, it is almost 1,500 times more sweeter than sugar. It is a protein, with the result actually it has some limitations you cannot use in a hot beverages. Because any protein, when you heat it more than 65 degrees, it gets denatured, and with the result you cannot use it, but for the cold items it is feasible. Glycyrrhizin, another sweetener which is coming from the plant. MAG is something which is popular and permitted by the FDA. Monoammonium glycyrrhizin. Okay, next slide please. Phytochemicals in the other sector, lawson, mehendi. I am working actually with the Godrej now, and the problem, of course, I don't dye my hair. But this big sector, 5,000 crore. For Godrej, it is a 5,000 crore product. And they approached to me actually because they are using henna leaf. And henna leaf is having the problem actually because you have to first make a paste and then apply it actually, and then you have to wash 2 hours TV. Correct, no? You have to wash for 2 hours TV. I mean, you have to keep that actually for 2 hours. So that is the problem. So they want actually whether this can be facilitated. So they came to me whether we can appreciate the quantity of the lawson to 20%, what it is present only 1 to 1.5%. So I developed actual technology for the 20%. Now I am ready even for the 95%. Natural lawson. Okay? And so lawson is the one dye which has been used. Then the hematoxylon, there is another dye which has been used, which of course is not in the Indian plant, but then there is another dye which has been used. So lawson is the naphthalene which has been used for that. Indigo Fera Victoria, this is actually the denim, it was used actually for coloring denim. Limonene currently is limonene is a byproduct of citrus fruit oil. Okay, and the important component from the perfumery angle is citral. Citral is actually a monoterpenoid, which is a combination of geraniol and the neral, that is used actually in the flavoring industry. But limonene, which is the major constituent in terms of the percentage, because almost 90% compared to limonene. Limonene is a hydrocarbon, and all hydrocarbon are good actually, but it's a biodegradable hydrocarbon, most important, unlike the one which is coming actually from the mineral source. And this has been used in the industry actually as a cleaning agent, because it can dissolve grease and all that very easily. So it is used actually for degreasing, because you use grease actually for the lubrication purpose, but after some time, after accumulation of the dust, that grease becomes stubborn and that hinders the passage. Okay, so to free them actually you have to replace that grease. To replace that, to remove that grease, you need actually limonene. So that is the solvent which has been used. Decay value, I don't know how many of you know decay value. Sorry, Kp value. Corributan value actually. So it is having around 67 value, which is the most suitable for the solvent. Menthol, number 1 coolant. Natural, number 1 coolant. Thanda, cool. And polo. I mean, Karanjin is used in the many agriculturalists actually they used a lot of Karanjin or the Pongamia pinnata plant extract has been used. Actually, Karanjin is a major constituent in that, which is actually pesticide. Sesamin is a pesticide synergist. Sesamin, which is a lignan derivative from Sesamum indicum. Quinine, all of you know quinine. For what? Huh? Anti-malarial? No, it is not used for the anti-malarial at all. It is gone out of order now. Okay? Quinine today is used actually for imparting bitterness to the liquor. Bitterness to the liquor. Okay, that is the use of the quinine today. And quinine is a standard bitter. If you refer how to find out the bitter value, it is always determined in terms of the quinine. Oxalic acid is used actually in the cosmetics. Next slide, please. Okay, derivatives of the synthetic analog. Next. Okay, diabetes. Yeah, morning when I asked for the tea or coffee, I was expecting the question with tea with sugar or without sugar. People like me, moment you turn 40 and go to the hut and ask for the tea. They will ask, whether it's with tea, with sugar or without sugar. Okay. And we have this drug today actually, metformin, sulformin and the bitformin. Okay. And then lifestyle, bibonide, then we have fenformin, phenyl bibonide, and we have a butyl bibonide. So they are the drugs which are used extensively today. And origin lies in a plant called goat's slew or Gallica officinalis. That is the plant which is growing actually in France, which belongs to the Fabaceae family, and it contains actually a synthalin A. Okay. Decane NN diagonidine. That is actually a compound which is found to be present in that. This is galligine and the synthalin both are present in the plant called Gallica officinalis, and this has laid the foundation for the development of synthetic anti-diabetic drug. Okay, these are all bibonide, and this is the guanidine which is found to be present in this, and this has given the idea for the development of this particular compounds. Next, this is about the quinine. So we have a quinine. This is natural product. Then we have these are all synthetic which came with the picture, whether it is quinathine, chloroquine, hydroxychloroquine, became very popular during COVID. Correct, no? And Indians have made huge profit out of it, I must tell you. So you never know which molecule will bounce back. So keep them. Okay? Amodiaquine, primaquine, mefloquine. And if you look at all the structures, one thing is very clear, all of them they have a quinoline ring in that, all of them, most important. And second, this is called the quinaglidine ring. And all of them they retain the quinaglidine ring also in some or the other form. These two are the essential feature for the activity of a molecule. And this has laid the foundation. When the quinine structure was made available, that has laid the foundation for the development of the synthetic analogs. Next slide, please. Well, I will skip this here, all of you know this. Natural artemisinin is neither water soluble, neither polar nor non-polar. So that was the problem. Taxol story also went the same way. Okay, so that is why you have a derivative which convert into an artemisinin and then make it either artemether or you make artesunate. Okay, so this hydroxy group, this is a OH which can convert into artemisinin and then modify this to make artemether or you make succinic acid derivative artesunate. Okay, then it is more polar molecule, this is non-polar molecule. Next. So it improves the onset of the action. Alright, this is about the cocaine and the synthetic analogs. Cocaine from the coca, erythrosalon coca, and that has laid the foundation for the local anesthetic. Okay, so cocaine is of course, this is a propane alkaloid, okay, which has been derived actually from the ornithine. Propane alkaloids are derived from the ornithine, and these are all the compounds which has come because this exists in the nature, the structure was made available, and that has led to the development of these compounds. Next, another important thing, all the steroids today, most of the steroids are obtained actually from tyrosine, and I think this is included in the syllabus now, the undergraduate. Okay, converting into diagenin, and a lot of them are finding it difficult because what I am supplying it actually is the entire tuber, and everybody is asking for the powder. It doesn't get powdered, and don't put it in the mixer. Mixer would break. Okay, alright, dialysine, it is present in the form of dialysine to the extent of around 4 to 5%. It starts from the 3%, and it has to be hydrolyzed first and then converted into diagenin, Diogenin then it will chemically change actually to the 16-DPA, that is 16-DPA pregnant acetate. This is pregnant derivative, which is a C21, 18, 19, 20, 21. And then of course it gives the formation of the different steroid, hydrocortisone, betamethasone, dexamethasone, which are used actually as an anti-inflammatory drug. Okay. Next, cathinone. Don't get into this. Have an idea, but don't get into this. Huh? They're all the party drugs. Okay. This is cathinone. Cathinone is found to be present. In fact, in Ethiopia, there's a habit like we have a betel leaf chewing habit. Like that, in Ethiopia, they chew actually a leaf of Catha edulis. That is Catha edulis, which they call it cat. Okay, and that contains cathinone. Cathinone, like those who stayed in the hostel, and before examination, you have to burn the night, and then you take amphetamine. Correct, no? Yeah. Because that will keep you awake. Alright. So this is about, so that is what actually when they used to go to jungle actually for getting the thing, so they used to eat this leaf actually and I don't know, hudia? Anybody knows hudia? Hudia? Okay, hudia is another, today is a compound actually, hudia is a plant in the, again Africa, it's a cactus and hudia garodiana, that is actually the plant. And many Africans, they eat this plant and they go to the jungle actually for killing pain. You know? And this particular plant keeps them actually energetic without consuming anything. And that is how it has been used today actually as an anti-obesity drug. Have you got it? Anti-obesity drug. Okay. Hudia, I think you will be able to get some idea on the Google. Alright, so Catha edulis is what the khat, and we have actually the ephedrine. Ephedrine don't, again actually a narcotic. In Bombay, we have a problem. Last 2 years, NCD, narcotic. Okay, all right, Sushant Singh. Okay, these are all the drug actually. Amphetamine, yes. Methamphetamine, that is N-methylamphetamine, methcathinone, so derivative of the cathinone. Ephedrone, again derivative of cathinone. We have the derivative actually of amphetamine. Okay, SKTC and of course MDMC. So these are the party drugs actually. Don't possess and don't use it. But yes, a good money is lying in this. Next slide, please. Okay. Yeah, important thing. Morphine and synthetic analog. This is the morphine which comes actually from opium. Opium contains around almost 40% of morphine in that. Okay. Morphine, codeine, noscapine, and thibane. Okay. Those are the major constituents in that. Morphine to heroin, a simple one-step reaction, treat with acidic anhydride and acetic acid. Okay, you get both the hydroxy group acetylated and becomes heroin, which is again potent, 2 times potent than the morphine. And that is the reason actually why you don't get the acetic anhydride easily. Okay, that is the reason. And then we have a fentanyl and sufentanil. Okay, these are the 2 more opioids which have been developed actually by looking at the structure of this. And if you look at this fentanyl, it is, some people say 100, actually it is more than 100 times more potent than the morphine. Okay, and sufentanil has not become, according to me, I think yet it has not become a drug, but fentanyl has become a drug. Okay, I must tell you another very important story which has happened because of fentanyl from ICT, one of the ICT actually he has developed fentanyl in the form of the spray. Okay, mouth spray, that means you put it in the mouth actually and immediately the pain will be relieved. And he got permission, this is about the US, okay? These are opioids, so opioids are under control now, okay? So this got the permission actually for making spray only for terminally ill patient like cancer, okay? But he became more ambitious and he started selling, because in US actually the thing is different, okay? We and me, in India we have a good capacity, tolerant capacity. When you suffer actually a couple of days, you try some household thing, correct, no? And then doesn't work out, then reluctantly go to 30 to the doctor. This is what. US is not like this. The moment you get pain, immediately they want painkiller. Get relieved, that's it. So he developed actually a spray, which became very, very popular. He made huge, in fact, fortunes actually. And in a process, he also dealt with some illicit activity of promoting, because he got permission only for selling to the terminally ill patient, cancer. But then he made money actually by selling this product, and now he's behind bar. Fentanyl. Okay. Fentanyl, so it is almost 500 times, more than 100 times more potent actually in terms of the pain killing activity as of morphine. But yes, all this has led to the development only because the structure of this was made available. Next slide. Any? Do you want me to continue or?
Yes, yes,
Okay.
yes. So
Next.
we have another 10 minutes.
Okay, fine.
Thank you, sir.
All right. This is about the thymol. I don't know how many of you know thymol derivative. This is the derivative WS3. Okay, which is derivative which has been used now today. In fact, WS is a company, Wilkinson Sword is a company actually, and they have used it. They have made the derivative for their cosmetic purpose. Coolant. Okay, and if you look at this, menthol lies here, and WS3 is having more cooling sensation than the menthol. So natural, menthol which has been derivatized. In fact, it has been converted to a paramethylene 3 carboxylic acid, and which is then converted into a ethyl carboxamide. And that is now 150 times, that is 1.5 times more coolant than the natural menthol. And this is altogether synthetic. This is of course natural menthol modified, and this is synthetic. Next. This is the project I just told you about. We have converted barbaline in situ to aloe. Okay, this is barbaline, which is converted to a aloe in situ. So we have said only technology for this. Then of course it will be converted to a diacetyl, that means carboxylic CH2OH group is converted to a carboxylic acid group oxidation, and then of course 1,8-dihydroxy is being acetylated to convert into a 1,8-diacetyl rhine, because this becomes rhine actually. Okay, this is rhine compound and this is 1,8-diacetyl rhine. That is called diacetyl. Diacetyl is the only drug which is figuring in pharmacopoeia today, which is targeted as an anti-arthritic drug. Okay, and this is the only source. There is no other source for getting diacetyl. Okay, it has to go from the barbaline to aloe to rhine. Some people are making it from here to stay like this, but otherwise this is the route for this. Okay, next slide please. Natural anthraquinone. These are the dyes actually which are used. This is of course rhodium, teak wood. We have a teak wood benches in our institute. And teak wood is very durable wood. The reason actually because it contains one naphthaquinone and one anthraquinone. Okay, that is why it is durable. Okay, next slide please. Next, I'll skip this. They are synthetic dyes of anthraquinone. So this is aluminum. This is matoxanthin, which is an anti-cancer drug, which is again anthraquinone. Rufigalol. Rufigalol, of course, can be prepared actually because antimalarial drug. Rufigalol can be prepared from the gallic acid, because if you see the structure, you can see there are two gallic acid here. Okay, it can be prepared from the gallic acid, dimer of the gallic acid. Then diacetyl next. Pyrethroids. All of you are using some mosquito repellent at home. Follow something of the kind, and I am sure none of you will be knowing actually what it contains. Okay, it contains either transfluethrin or dialethrin. Okay, these are the synthetic analogs actually which are prepared actually from natural pyrethroids. And if you look at the natural pyrethroids, it has a two important features in the structure: dimethyl 1,1-dimethyl cycloprotein ring, and you have a ester linkage. And if you look at the synthetic analogs, they have maintained these two features in that, then only the compound is having the insecticidal or mosquito repellent activity in that. Most of the mosquito repellent at home, it contains around 4% alethrin or transfluethrin in that. Next slide please. Next. Well, as I told you, you need phytochemical for the evaluating actually the both qualitative and the quantitative. Okay, next. This was actually one project which I got it from the Singapore, and the challenge was actually they wanted to show whether all the 9 drugs are present in the formulation or not in the capsule, and if they are present as per the label claim which was made on that or not. Okay, so they wanted the proof of this. So what we did, we took out one marker actually from the each plant. First we demonstrated the identity of that means by the TLC. Next slide please. Yeah, TLC. So all those have been demonstrated by the TLC. There's 8 different formulations. So in that we have shown actually that particular. This is astagaloside because the major plant which was put into that. So the qualitative analysis, yes, and then the same thing was also shown actually with the help of the HPLC. Okay, so that was about it was a very difficult task because I don't know how many of you are running the HPLC of the natural product, because in HPLC when you started actually, once the pointer goes up, it doesn't come down. It just remains actually up. Okay, that is why you need to be very careful actually. In fact, you need good amount of preparation of the sample. You need actually, because preparation of the sample without compromising the constituent of the analyte you need to prepare a clean sample, then only you will get the peak, which will be starting and coming down to the baseline. Otherwise, once it goes up, only when it completely get exhausted, it will come down. No? All right. So this is a very difficult task actually of coming back to the baseline. People draw all that things, I mean, it's all slanting. Next. Okay, this is one patent actually which was granted actually by the US patent, and the thing was actually because we were using 9 herbs, and some of them were water soluble, some of them were oil soluble, the non-polar, and we used the solvent actually which was combination of the alcohol and the water, and then the challenge was actually whether all those herbs which are there, whether they're getting represented or not. So we have here actually track 1, standard amygdalin, and then we have the 3 tracks. One is the water extract, water cum alcohol extract, and then the alcohol extract. Okay, and in every case we have shown actually this is the amygdalin, which is of course a glycoside, which is found to be present in all. I mean, in water, yes, it is more. It is there in the water alcohol mixture, but in oil mixture, I mean, sorry, in the only alcohol it is less. This is about hydroxy safflower dye, which is more water soluble. So, in middle track, actually, this track is of the alcohol and the water mixture. So, there you can see the strong presence. Okay, in alcohol it is not, but in water and alcohol it is more, but in water it is less. Once again, yeah, last one is the water extract. Sorry, this is about asaron, beta asaron, which is present actually in a Indian plant called calamus, but they are using the different variety called Acorus tatarinobi. What we have actually Acorus calamus, okay, tatarinobi, and that is not water soluble. So in water you don't get the presence of it here, but then I think there is some problem actually of the. It is found to be present in only alcohol and the water mixture, and based upon that we have quite a patent it was granted also. Next slide, please. Next, I will quickly go through some of the. Yeah, next. These are all the. This was generated actually for the quality standard of Indian medicinal plant. Next, I'll skip this data. Okay, this is the Lawson story, and we are still battling actually for the filtration problem, but yes, we are used, but alkali actually very quickly extracted. Next, yeah, this is about the extraction of the hinari powder with the weak alkaline water. Then of course it was neutralized and extracted in the non-polar solvent, and thereafter of course it was purified actually once again by because it's a leaf. So when you extract the non-polar solvent, the amount of chlorophyll also comes along with that. So that was the major problem which was coming in the way. Next, kaolin phenolphlemonide, phenochromone rather, sorry, not flemonide, phenochromone derivative, it is not flemonide. Next, this is the kaolin, the extraction, the isolation. Once again, this were used directly for the ICMR monograph. Next, okay, skip. Alright, this was recently we have developed the method actually, and it was captured by the wiki, and it was put actually as a reference. Next, Magiferin, okay, Magiferin indica or Bombax saiba. Go back to the earlier slide, yeah. Bombax saiba. So Bombax saiba is also known as the kapok. Kapok is a sour, in Marathi it is called the sour. Okay, sour and Kateri sour, there are two plants actually. And Bombax saiba also contains Magiferin. Magiferin is a xanthone derivative. This is xanthone, and it is present actually more in the mango bark rather than in the leaf part of it. But leaf also contains Magiferin in there. It is actually an antioxidant which has also become popular as a nutraceutical product. Next, xanthone derivative. This is the product. This is the nutraceutical product. Next, these are all confirmation of that. Next, it obeys that Shinoda's test. Next, these are I think I already told. Next, okay, go ahead. Lupiol. From Platyva murvala, that is a plant, yeah, Platyva murvala or Platyva magna mount is a new name, and this is from the Capreaceae family. Caper is a, I don't know, kale, anybody knows, Rajasthani vegetable, sangri, kale sangri. Okay, kale is actually the referred to the fruit of a Capreaceae, aphylla. So it is belongs to the same family. This is the family which normally contains sulfur in that. Okay, glucotropylene, those kind of constituent. And the bark of this was captured actually from Jijamata garden. Okay, there is a tree of Platyva murvala there, a religious plant normally it is used, and it contains lupin derivative, tetrapenoid, lupin derivative, and currently lupiol is also used in some cosmetics. Next, so we have developed method for that. Next, tetrapenoid. Next, just confirmation of that. Another tetrapenoid, ursolic acid. Ursolic acid is present in many plants like Indica rosea, tulsi plant, and currently ursolic acid is used actually as one of the marker compound for tulsi extract. Tulsi extract should have the two components. One is a tannin, and second one is a ursolic acid, and third one is volatile oil. But most of the water extract do not have that much of the volatile oil. But two constituents they wanted, tannin as well as ursolic acid. Normally, it is standardized to around 1 to 2% of ursolic acid. And for that, in fact, some people they also took ursolic acid to spike. Understood? To spike. If you are not getting it, spike it. Next. Alright, this was obtained from the Nerium oleander. That is actually a Nerium oleander plant. Kaner in Marathi. Next. Next. Only problem with the plant actually it also contains oleandrin, which is actually a toxic compound. It's a cardiac glycoside, which is a toxic compound. This is about the phosphorine. I think I have already narrated you the story. Next. Next. Yeah. It acts on the adenylate cyclase enzyme, which in turn improves the cyclic AMP level in the body, and that is how it is related to the obesity. Yeah, and this. Next. Karanjan. Go ahead. Karanjan is from the Karanja. I think in Bombay, every third or the fourth tree is a Karanja tree. Okay, in Bombay. So Karanja is very common tree which is being planted, and it is also having, Maharashtra is also having a huge forestation of Karanja. Okay, these are the seeds. Karanjan, I'm isolating Karanjan actually from the, it's again accidental. In my company actually, I'm dealing with the Karanja oil. And once it just happened actually, I have purchased around 200 liters of the Karanja oil. And it was lying for 6 months or so as it is. And after 6 months, I could see actually a nice layer of sediment at the base. Okay, and as I told you, I am quite interested in finding out what it is. Always so. So I took that actually and run the TLC, and to my surprise, actually it came out to be a reach in Karanjan, and that is why I started separating the Karanjan from that. Okay, so this is a Karanjan which has been separated from the oil. Karanja oil has been used actually for the separation of that. Next. This is one Igdasone story. Hydroxyidasis is a phenomena of shedding the skin or feathers. Okay, that is the phenomena actually, and this particular compound is having the importance in the silk industry. Next slide, please. Okay, this is a very important. This is the life cycle of the silkworm, Bombax mori, and these are the eggs. Now it has to from larva to the pupa stage. This is the pupa stage and this is the larval stage. So larva to the pupa, there are the 5 instars. 1, 2, 3, 4, 5 instars. It has to undergo 5 instars. At every instar, it has to shed the skin and grow. Unless it sheds the skin, it cannot grow. Okay? And this particular phenomena is controlled actually by the Igdasone as a steroid. In normal case, they get this steroid actually from these mulberry leaves. Okay? But then the typical problem which is being faced by the farmer was actually that the insect used to mature at a different time. Some are maturing today, some are maturing tomorrow. So for small farmers it was a problem because every day they have to go with the produce to the market. So they wanted some uniformity. So what we did, the company came to us actually that they wanted 15% Igdasone so that they can sprinkle it actually on the leaf so that all the insect will mature at the same time. And that is what we did. So these are the 5 instars which typically last from 3 to 8 days. And during this period they have to shed the skin. Then only they start spinning the cocoon around it. That is actually the source of the silk. Next. So this is what we use Caladana, that is Apodia hediracea. We belong to family Convolvulaceae as a source of the Plantaria, and we could give actually more than 15% Igdasteroids, not Igdasone alone. This is 200 hydroxyigdasone. We have given the Igdasteroids, which is a combination of many Igdasones. Next. These are all the Igdasones which are present in that. Next. This is just confirmation. Next. Yeah, this was the large scale process which we undertook actually in the company. Next. These were some field trials. Next. Yeah, this is the part which we came out eventually from this Sampurna. This is the vial which was to be dissolved into the 20 liters of the water and sprinkle it actually on 500 kg leaf. Okay, on which so the other field trial which we are undertaking in the Bangalore. Next. Yeah, this is tomatin, another important case study of extraction isolation. And this project I have undertaken in the Sharjah. And because the product comes actually from Africa, we don't have this particular plant growing in India. And they were processing at Sharjah. The typical problem, next slide please. Tomatococcus daniel. The typical problem, these are the fruits actually, get up the fruit. And from the aerial of the fruit actually it is obtained. Aerial seed surrounding to the seed actually, that is the aerial part of it which has been used actually for isolation of the tomatin. And thaumatin is a protein. So protein handling is very, very difficult. And our batch, typical batch used to be 5 kg batch. And for 5 kg we were using 1200 liter of the buffered water. So you can imagine the quantity of the water which we are using for 5 kg, 1.2 ton of water. And we had a difficult problem of the filtration because this fruit, like any other fruit, contains a lot of mucilage. There was another option, you cannot use any solvent because it is a protein, so it will not dissolve in any solvent, so you have no option but to use water as an extracting. And we had a difficult period actually for filtration because a lot of mucilage was there and that was coming in the way of the filtration. And filtration was because of the mucilage. So what we use the chelating agent for the mucilage. I will not be able to reveal that compound, but we have used that chelating agent by which mucilage is complexed, and then the filtration became much more easier. Okay, it has undergone first the microfiltration and subsequently actually with the membrane filtration of the compound was of the 21 kilodalton, whereas we were using actually 5 kd membrane. So all other constituents used to go actually the retentate. We used to undertake this, freeze drying first, freezing actually, and thereafter the freeze drying. So from the 5 kg, we used to get typically around 600 to 700 gram of the thaumatin. Current rate of the thaumatin is 2.5 lakh to 3.5 lakh per kg. That is the rate of the thaumatin. Okay, next slide please. Yeah, this is about the 5 kg jar where you can barely accommodate around 600 to 700 gram. Next, major challenges with mucilage. Well, these are the phytochemicals we have displayed in our institute actually isolated. This is the group which I was talking to you about, where we have come up with the 25 case studies. Next. These are some of the technologies which have been transferred to the industry. Next. ICMR volumes. These are the chocolate which I have just mentioned to you about Erlang as a company, so they have come up with the chocolate. 2 bar and these two are actually gummies. Now we are coming out actually with the candies. Okay, one for the sleep and the one for the anxiety. A major problem. Huh? Okay, these are some cosmetic products. Arrow juice. I'm having. I'm associated with the Pidilite for their CSR activity, and one of the CSR are manufacturing actually the aloe vera product, wherein they are making juice as well as a gel part of it. Next. That's it. So technology development for extraction and isolation of the phytochemical and their effective application will lead to the development of rational phytomedicine. I have used the word rational phytomedicine, okay, which will have the global acceptability, not just regional or the Indian, but then we must make the product from the global angle and not from the regional angle. Thank you very much. Next slide, please. Once again, sincere thanks to all participants, AICT, I think the ATAL, because the sponsoring agency, and Principal Madam, of course, all the participants who have participated for this particular FDP. Thank you once again. If you have any question, I will take it. I'm okay. No issue. I'm used to working in the lab.
Request you to be please seated. So please be seated. So at the outset, I would like to thank Sir for deliberating a very elaborative and a very inspirational talk on botanicals and the challenges and opportunities that are involved with. Sir has given us a very beautiful insights on how we as academicians should look at the unmet needs, identify the problems, and then give those problems to the research students so that even they get into it, they get into the depths of it and they understand that why they have to work for an industry-oriented solution. Sir also explained on the concept of how should be the approach of the research guide when we are working in a laboratory. What kind of focus should be there on, first of all, identifying your niche area, identifying the challenges and the problems that are associated in that area and then working on it. Sir also mentioned about how we as academicians can bridge the gap between academia and industry by constant interaction with the industry, meeting them, understanding their problems, and then working on science and technologies which will at least bring down that problem to a certain extent. Then Sir also took us through a number of case studies. Remarkable ones were on the synthesis of various phytoconstituents which are are known as major phytoconstituents like morphine, then quinine, cation, menthol, and so on. And very importantly, the message that Sir has given all of us from this particular session is that it's very important for us to have a self-audit every day. That what is your outcome in terms of your contribution when you are contributing to a day's work. We have to spend out some time at the end of the day to look at what is our outcome for that one particular day in our lab. And most importantly, always focus on the science and technology that you have learned, because only then you will be able to develop rational phytoconstituents when it comes to the area of herbals, and rational processing, rational solutions when it comes to in whichever area you are working. So sir, on behalf of BS College of Pharmacy and all the participants, I would once again like to thank you for being with us here today. And now I request our principal ma'am, Dr. Supriya Shide, to felicitate sir with a token of appreciation.
Send all these photographs to the
Thanks, sir. Very informative.
Participants, the session is open for questions. If you have any questions for sir, then please go ahead.
Yes.
Thank you so much, sir. First of all, I am feeling so fortunate and lucky that I'm attended and here present today to listen to you, because a lot of knowledge regarding these constituents and this medical rationale and how it is being applied in the industry. It's fantastic. I would like to know about some diseases which are really neglected and yet not that sufficient research is being done on that. For example, vitiligo. Okay, so I would like to know that any plant extract, because recently that only one drug that is got FDA approved, sorry to name it not. But is any medicine plant extract can be work out for this vitiligo?
Vitiligo normally, vitiligo is actually your melanin.
Yes.
Okay. See, that is deficiency of the melanin in the skin, and PUVA is a treatment. PUVA is a treatment for that. That is actually Soralen come UV rays. UV rays will belong to the A range. That is that starts actually from the 320 or 342 or the 400 to 320. That is the range actually. PUVA treatment which is normally being indicated. Soralen, methoxylane, these are some of the Soralen is actually a natural product. Soralen is a natural product actually which comes actually from a bauchi. That is the Soralia corylifolium. Okay, but then the same compound is also phototoxic compound. So that stimulates the formulation of the melanin, but at the same time if you take it long, more quantity, it will also lead to the development actually of the redness of the skin, and it can go further. It can complicate further. So one has to be very careful. They are all coumarin contained, coumarin plant material, those having the capacity to stimulate the melanin formation. Yeah, they have a capacity, but one has to be, and this should be used only under medical supervision because they are also phototoxic compound. Quite a few, even citrus fruit like our Eagle marmalose, bale also contains actually a marmalocin or imperatorin. Marmalocin is the imperatorin, which is actually a phenylcoumarin. That is also having the same activity. Both root as well as fruit contains actually imperatorin in that. So all coumarin, for that matter, coumarin and furanocoumarin, they all have that melanin stimulating activity, but yes, only when they are subjected to the UV irradiation. That is why normally doctor will ask you to take this particular drug and then expose yourself actually for some time to the sun rays. Yeah.
Thank you, sir.
Anything? Yeah, tell me.
So from the expertise and technical knowledge that you have, what is your say on Amara as a anti-diabetic, which is so much in hype with the powders coming up, and you take 1 spoon with water, or you have some wood pieces which you just put it in water and drink that water in the morning.
I thought that I will keep the session scientific. But now you are taking me to the emotional. See, a lot of wrong things are going on in the society without scientific. Like morning I said, when Amazon Ashini comes, and that is a quassia, okay? And but what happens actually in the society? Anything which is bitter is considered to be good for the diabetes. Karela, bitter, good for the diabetes. We don't know, okay? So likewise, quassia is a bitter, okay? Anything is bitter is diabetic. Anything which kills the bitter, which kills the sweet taste is also considered to be diabetic. Kills. Gymnema genin. Gymnema, if you take 1 leaf, okay, if you chew 1 leaf actually for next 2 hours, whatever amount of sugar you eat it, you will not perceive the sweet taste because it is called the anti-sweet. Now, if you call anti-sweet as the anti-diabetic, I will leave it to you to decide. More pharmacology should be done actually, and but yes, there are quite a lot of things of this kind of thing like look-alike, having the similar kind of things are existing. What we need actually, we need to scientifically validate those claims, and for that the pharmacologists must come forward and do their job. See, one should take it as a clue, something which is already in the folklore or in the some treatise which has been mentioned. I think we should not just, I'll tell you, I don't know how many of you referred actually, Kirtikar Basu, Indian medicine planned by the Kirtikar Basu, very voluminous. They were kernel, okay? They just compiled the data on medicinal plant. Every plant is good for the snake bite, okay? It just so happened actually that, some villager actually snake bite has taken place. Okay, so he has taken some leaf, applied actually or chewed, and he got cured because the snake was not poisonous. Okay, but the credit went to the plant for nothing. Okay, today we came to know that not that every snake is poisonous. So that is how the thing went. We need to correct that. And that will happen only with the scientific validation. That is what we must do, that actually such claims always. See, Kekule when first found out when he dreamed about the benzene structure, what was the structure? Snake. That one we put 3 double bond in that. Correct, no? That one we put 3 double bond in that. So this is how it will happen. So those things need to be corrected. And that must be corrected by if not by one model, you take many models and see if it is working. Make it put it in the usage. But if it is not working, at least validate with some different models and make sure that yes. And scientific community must come forward. Most important. We failed many times. I don't know how many of you heard about the herbal petrol. Herbal petrol, and one gentleman was associated with that, and he got pushed. Initially, he was given jail plus security, and then he was put behind bar. That was the story. Lord Ganesha drinking milk has happened in India and in Mumbai. Okay, because scientific community failed to come forward and explain that. My wife also started the same thing, that Ganpati is drinking the milk. I said, do one thing, take a vati, put a milk in that and put the Ganpati also and see, monitor whole day. Whether the milk level comes down, it doesn't come down. Okay? So that is how we must come forward. The scientific community must come forward. Yes, sir.
Sir, I have one question. Thank you so much for informative session. Can you throw some light on the patent protection for herbal products? Are you protecting the derivative of phytoconstituent or the process of extractions? Because we have one US patent got granted.
Phytoconstituent cannot be patented because they are there in the nature. They cannot be patented.
So the traditional lamate is not patented?
No.
Okay.
What you can patent? The process. Process you can patent it. Application you can patent it.
And have you licensed that patent to some other companies, any client?
In fact, this patent I have not paid even a single pipe. So it was patented by the company where I happen to be inventor. Okay? So my gain actually, the only gain is I have not paid even a single pipe, that is my gain. Otherwise, I don't know whether you know, what is the amount of the money which is required to file a patent in US. Okay, so that is my saving.
Thank you.
It was an industry project actually where I had no control because I have, see, whenever I work with the industry, normally what I practice normally, I surrender 100% to them. Okay, because they invest the money, so it is their property. Okay, I only get actually advantages. I get actually advantage in terms of that it is in my name, and some of them are ethical, very ethical. This company is very ethical because it was France-based owner, and companies in, of course, in Singapore. And first they have sent me to the China actually to study the entire process, and the entire process was of the 6 days. And he asked me whether the same process can be shortened. And then I got into the action actually, and then we did a lot of batches in our laboratory. We had a good project actually, almost to the extent of around 80 lakh rupees project. Even during COVID actually, I had a project on that. And during COVID, I was fortunate actually because I had stayed right behind. I was able to work every day. And during that COVID project, I had a project one from the Godrej, another from the Singapore-based company called Molia. So I kept myself busy during COVID, plus developing some technology for the extraction isolation. Yeah. So you can have a patent actually for processing.
I would request the participants, sir, in honor of time, please kindly post your questions to us and we will definitely pass it on to sir.
Yeah. Quick question if you have.
No, sir, I just wanted to say that silver which we use, milk thistle plant.
Milk thistle.
Other than that, any other source?
No. Milk thistle is the only source. Silabum marianum.
It's in China.
Yeah. No, it's Europe also. Entire Europe. It's a northern hemisphere. This plant is actually from the temperate region, so entire northern hemisphere you get actually Silabum marianum. I mean, people should try it here also. It can grow in Himachal Pradesh also. I'm sure it can grow. Asteraceae family or the Compositae family.
Thank you so much, sir.
Thank you once again. Thank you to all.